In this lesson, we are going to talk about what happens when chromosomes are not, or genes are not on different chromosomes. Because as you'll recall, chromosomes have lots and lots and lots and lots and lots of genes on them. A gene is a tiny little section of DNA that codes for a protein. And a chromosome could be made up of hundreds or thousands of those depending on the size of the chromosome. And so in real life, genes are not always going to be on two separate chromosomes. So we're going to talk about something called chromosome mapping. Now, just a bit of context. There is a theory called the Chromosome Theory of inheritance. This chromosome theory of inheritance can be linked to Morgan, who we mentioned before. And so I'm just repeating his name because he came up in the last unit. He's the one who said genes are on chromosomes. But then there was another guy, his name was Walter Sutton. He was studying grasshoppers. I don't know about any of you, maybe when you were kids you did this. My daughter and her friends like to go to the park by our house and trap grasshoppers and then put them in containers and then, I don't know, do what with them. I always tell her she has to let them go at the end of the day, but I'm not sure that they do. So Walter Sutton was studying grasshoppers. Often people study small organisms because they tend to reproduce in more or in larger quantities and more quickly. And so they're good for studying genetics because you can have a lot of offspring produced in a relatively short amount of time. Now, he agreed with a number of things that we have already said. First, he said, yep, gametes follow Mendel's model. Mendel said gametes are produced, and when that happens, segregation happens. And so that brings us back to the idea of meiosis. During meiosis, I start with a diploid cell. I end up with haploid cells that have one of each chromosome instead of two. He agreed with Morgan that genes are located on chromosomes. But here's the point that really was different. He realized that genes that are located on the same chromosome do not always assort independently. Notice I didn't say do not assort independently ever. I said they don't always. Sometimes it kind of seemed like they did. And so we're going to call these genes that are on the same chromosome linked genes. They're on the same chromosome, which means they're inherited together. Now, you're going to notice that we're not going to make Punnett squares today, and that's because predicting the outcome or the percentages in offspring when genes are linked is not a typical question that we would ask of a Biology 30 person. What we would want you to be able to do is to recognize that if genes are on the same chromosome, they are linked. They will get inherited together. And so if I said to you, hey, the gene for eye color and hair color are on the same chromosome, then you would interpret, well, I'm going to inherit those two characteristics together. Now, the thing we're actually going to focus on is how these linked genes could potentially separate. Because, remember I said, they don't always assort independently, but sometimes it kind of looks like they do. The way these linked genes can separate is because of crossing over. So remember back in the first unit when we had a pair of homologous chromosomes, each of them had a sister chromatid attached by a centromere. And in prophase 1 of meiosis, the ends of two chromatids could cross over each other when synapsis occurred and a tetrad formed. And so this is a specific way that genes that are on the same chromosome could end up separating from each other. Crossing over could occur, and the version of the gene on one chromosome and the version on the other could kind of trade spots. It occurs more at points that are at the ends of the chromatids. That makes sense. It's more likely that the crossover happens with the tips of the chromatids rather than where the center of things is. So what we're going to practice doing is using some data about crossing over to try to figure out where a gene is on a chromosome. That's why the title of this lesson was Chromosome Mapping. Now this is just the context, the background. Does anyone want to ask anything about this so far? So, I'm going to show you a little bit about chromosome mapping, and then we'll do the first couple questions out of your practice problems together so that you can see the strategy that we use. Now, there's a few more definitions that come at the beginning of this little explanation. The first one is a recombinant type. This is what we would use to describe F1 descendants, so offspring in the first filial generation, that have different combinations than the parental generation. In other words, a recombinant type had some crossing over occurring. And the crossing over led to the offspring having the genes assorted in a different way because of the crossover. Now, crossing over doesn't happen and cause change all the time, every single time. And so there will also be descendants in the F1 generation that we would call parental types, where they have the same combination of genes as the parents, and we would infer that crossing over did not change anything. And so what we would want to do to give us some data is to figure out, of all of the offspring in the F1 generation, what is the frequency of recombination? In other words, what percentage of the time did a crossover actually take place? So we're going to look at the parents, look at the offspring. If the offspring have the same combination as the parents, they go in parental types. If the offspring have a different combination than the parents, they go in recombinant types. And we'll figure out what percentage of the offspring are those recombinant types. It's usually given as a percentage. It could be given as a fraction or a decimal number too, but percentage is really the most common type of data that's used. So, once I have this data, once I figure out, okay, I have this many offspring and this percent of them are recombinant, what on earth do I do with that? Well, I'm going to take this percentage and convert it to something called a map unit. Scientists are great at this. We make up units to suit our purposes. When we're like, the thing I want to measure is really small. Let's make up a new unit so that I don't have to use decimal numbers. Like a mole, you guys remember that in Science 10? The size of atoms is so small that to talk about one atom is silly. So we made up mole to talk about an amount that we actually want to discuss. A map unit, or MU, will be equivalent or directly proportional to the recombination frequency. In other words, 1% recombination will be equal to 1 map unit. So let's say that this thing right here, I'll make it yellow so it's a different color, this is my chromosome. And I determine that the gene for eyes and the gene for the body shape and the gene for wings, these are all referring to fruit flies because we study them all the time, are located at those positions along the chromosome. How would I have done that? Well, if you notice, I indicated that there are 4 units between the gene for eyes and the gene for the body type. That means that there was 4% recombination between those 2 genes. And that's the data that you would be given. There was 4% recombination between the gene for eyes and the gene for the body shape. So you would interpret, they must be 4 spaces apart on the chromosome map. Between the gene for body and the gene for wings, I indicated there were 6 units. Clearly, this is very well drawn to scale. That would mean that between the gene for body and wings, there was 6% recombination. So 6% of the time, crossing over happened between those 2 genes. Since it was 6%, I put them 6 spaces apart on my chromosome map. Now the other thing that's on the map is 10. Even if I hadn't told you that there were 10 units between the gene for eyes and the gene for wings, you should be able to interpret that because you know how far apart the other genes are. So even if I hadn't told you, you should be able to tell me that based on this, there should be 10% recombination between the gene for eyes and gene for wings because there was 4 and 6 between the other 2. So our goal in this lesson is going to be to create a chromosome map like this one. So I'd like for you to take out your practice problems and we're going to work through a couple of them together. So I've just copied and pasted the first 2 questions from the lesson 3 practice problems over onto this slide so that I can show you the strategy that I would recommend you use when we're talking about chromosome mapping. The first question tells you what the frequency of crossing over between several different genes is. This is the typical type of data that you would get in a question. We'll give you a bunch of percentages. So my strategy, whenever I'm looking at a question like this, is to find the biggest number first. Now once I find the biggest number, the biggest percentage, I take the 2 genes that have that big percentage of recombination and I put them on opposite ends of my chromosome map. So it said E and G had 10% recombination. So on my chromosome map, which I tend to draw kind of like a number line because I'm not a super great artist and I can draw lines if my paper has lines on them. So I would represent that E is on one side and G is on the other, and that there are 10 spaces between them. Now, when you're representing the spaces between things, you could draw, like I did there, numbers in between each letter. But you might notice, especially when we do number 2, there are lots of letters, and that can get a little clunky, a little confusing. So what I might suggest is that instead of trying to do that you treat this like a number line. I would call this spot 0 and this spot 10. Then I don't have to write this in between the genes all the time. I can just put little ticks for all of the numbers in between. Now, I put E on the left and G on the right. Did the question say that that's how it was? Nope. And I could just flip this chromosome over and it would still be correct. So, the fact that I put E here and G there is just because that's the way I read the question. You could very realistically have drawn this same thing by putting G here and E here. It's the same response. What we're trying to do is figure out relative to each other where these genes are on chromosomes. And if that's the case, it usually means that there's potentially more than one correct answer for the order of the genes on the chromosome. Now I've placed E and G on the ends because they had the biggest frequency between them. Now I'm going to consider the other data. My next step is always to take one of the genes that's given and see if I can compare it to both of my endpoints. So in this example, here's F, and I have data that compares F to E and F to G. So I can compare this gene to both of the genes I already put on my chromosome map. That's what I'm aiming for. The reason I want to compare it to both ends is because I want to know, is it closer to one end than the other, so I can place it correctly. According to this, F is 6 map units away from E, but only 4 map units away from G. So, 6. So I would put F here, 1, 2, 3, 4, 5, 6 spaces away from E, and then I would check to make sure, 7, 8, 9, 10, that it was in fact 4 spaces away from G. And if I check, well, that's 6, and that's 4. So all of the data fits. If I had drawn my chromosome the other way, I would put F right here. And so there would be 4 spaces between F and G, 6 spaces between F and E. So if I asked you to make me a chromosome map, the top or the bottom would be a correct response because I didn't indicate what was left and what was right. And remember, I could just flip the chromosome over and it would still be correct. So my strategy was find the biggest one and then compare genes to both genes that I already placed and make sure that the numbers add up. Now the first one was pretty straightforward because there were only 3 genes. The next one has a little bit more data, and the percentages have decimal numbers in them, so it starts getting a little harder. But does anyone have questions about this first one? Yeah?
Is this on the quiz tomorrow?
No,
Okay.
on the quiz. Okay, so let's look at the second one. In the second one, I have all this data that Thomas Morgan collected with Drosophila, which is the scientific name for fruit flies. They could have bar eyes, carnation eyes, fused wing veins, and scalloped wings, and all 4 of those genes are on the same chromosome. Now if I take a look at my numbers in the second question, here's my biggest number, 11. So I would start my chromosome map by placing C on one end and S on the other. Now I put C on the left just because it came first in the question. If you want to put S on the left and C on the right, that's also correct. Then my next step, after I found my biggest number, is to find something that I could compare to both ends. So find another gene where I know how far it is away from C and how far it is away from S. Now I actually have more than one choice here, but I'm going to pick this guy, the fused wing veins gene. I can see that the percent recombination between that fused wing vein gene and C is 3, and between the fused wing vein gene and S is 8. So I would put 1, 2, 3 spaces away from C, FV, and then I would check to make sure that it was in fact 8 spaces away from S. If that's at position 3, then it will be 3 spaces away from C, and it will certainly be 8 spaces away from S. So that data works. Especially when there's a lot of data, I like to highlight or cross it out as I've looked at it so I know that I've considered it. Next thing I want to do is take a look and see if there are other genes that I need to place. it looks like I need to put this B gene somewhere. Now, I actually have more data than I need to place this B gene. And instead of comparing it to the two ends, I'm going to compare it here to F, V, and to S. Just to show you that you don't always have to compare a gene to the ends of the chromosome. As long as you have 2 points of data for a gene, you can compare it to 2 things. You should be able to place it on your chromosome map. So in this scenario, the B gene is 2.5% away from FV and 5.5% away from S. So when I look at that, I actually have 2 choices. I could put B over here, or I could put B over there. When I look at the numbers, I would say to myself, based on that, it kind of looks like, okay, thanks. It kind of looks like it should be on the right. If you're unsure, you can always try, but what you'll see is if you try putting it to the left, the numbers won't add up. So I'm going to put it to the right. It said it was 2.5 spaces away from FV, so 1, 2.5, right here. So it's kind of at 5.5. If that's the case, it would be 2.5 spaces away from FV, and from 5.5 to 11 is 5.5. So those pieces of data work. I didn't even need this last piece of data, but I would still check to make sure that it's correct. Is B 5.5 spaces away from C? Well, yeah, on my number line, it was at spot 5.5. So all of those pieces of data line up. So this would be my chromosome map. And so your goal in all of the questions for this lesson on the practice problems is to make a little chromosome map. Now, before I say a few more words, does anyone want to ask anything about these first 2 examples? Okay. So I just want to show you a couple of things on the practice problems. Number 4 and 5 give you the data in a slightly different format. Instead of just giving you percentages, they're showing you the number of times it happened, but the data is always for 100 eggs. So when I look at number 4 and number 5, and I see those numbers, since my total is 100, I know that those are really percentages. So where it says a 5 here, it means 5 out of 100, so it really means 5%. The way I would interpret that 5, because it's in the box where X and W meet, that means there was 5% recombination between X and W. So it's giving you the same information, it just looks a little different. So that's how you interpret 4 and 5. You'll also notice that there are some question marks. And it says in the instructions at the beginning, if there's a question mark, I want you to figure out what number should go there. So you're going to figure out, once you make your chromosome map, how far apart those 2 genes are, and tell me the number. Now, if we move ahead to the sort of end of this, just erase my thing here. Question number 8 is a slight bit trickier than the rest. In question number 8, my strategy of find the biggest number and put it on the 2 ends of your chromosome map will not directly lead you to the correct answer. And so I just want to remind you that that was just my strategy, a way of arriving at the answer. But then in number 8, you might need to do something different to place everything so that the numbers all add up. Number 8 is hard. It's harder than the average question, and it's probably harder than a question that you would be asked on a test. But I like to make sure you've seen it all so that you know what to do. Now, if you have any questions, I would love to answer them. The other question you should make sure you consider is from page 609 of the textbook, number 1. It's a question asking you not to make a chromosome map, but to think about what exactly we're measuring when we get that data, just so you don't forget. Now you have some